Hello everyone, I'm Susan. Today I'm going to talk about Ebola virus. So let's look at the West African Ebola virus epidemic first. It happened during the period of 2013 to 2016. It is the most widespread outbreak of Ebola virus disease, and it mainly happened in the countries of Guinea, Liberia, and Sierra Leone. It caused significant mortality, with the case fatality rate reported as slightly above 70%. As of May 8, 2016, the WHO reported a total of 28,000 suspected cases and 11,000 deaths. So here's the map that shows the outbreak by the time of November 2014. The darker the color that's shown in the map means that area has more cases. There are 5 species identified for Ebola virus. There are Zaire, Bundibugyo, Sudan, Reston, and Typhorus. These species all have capability of causing disease, but differ in extent of progression and virulence. The fatality rate varies from 40% for Bundibugyo, 50% for Sudan, and 70 to 90% for Zaire. All 3 of them are associated with the outbreak in West Africa. Ebola virus has a striking filamentous structure, which is shown in the picture. The viral envelope contains spikes that's consisting of the glycoprotein trimer, and this achieves the combined function of attachment to host cells. Fruit bats are the natural reservoir of the virus. It can be spread by saliva, such as kissing or shared drinks, or by touching a contaminated surface, such as blanket or doorknob, or by insect bites or stings, and finally it can be spread by blood products. So here's a picture showing the life cycle of Ebola virus. Ebola virus is introduced into human population through close contact with the blood, organs, or other body fluids of infected animals. The virus first attacks and invades cells that alert the body to infection. By doing this, the virus can evade the immune system and begin replicating itself. Then infected cells rupture, releasing more virus particles into the body, as well as a flood of cytokines, which are molecules that cause fever and inflammation. This cytokine storm damages blood vessels and causes internal bleeding. Certain white blood cells, called neutrophils, can even act as carrier and spread the Ebola virus throughout the body. The length of time between exposure to the virus and the development of symptoms is between 2 and 21 days. Between days 7 and 9, people can experience headache, fatigue, fever, and muscle soreness. And by day 10, people can experience certain high fever, vomiting blood, and passive behavior. Death, if it occurs, follows typically 6 to 16 days from the first symptoms, and it is often due to low blood pressure from fluid loss. Recovery may begin between 7 and 14 days after the first symptom. Survivors develop antibodies against Ebola that last at least 10 years, but it is unclear if they are immune to repeated infection. Now let's look at some detection methods. Most of them are rapid blood tests for diagnosis purpose, such as ELISA and qPCR. So ELISA is to detect a novel monoclonal antibody caused by the nuclear protein in the Ebola virus, and qPCR is to detect RNA of the Ebola virus within 3 to 10 days after the onset of symptoms. It is also quantitative. There are also some nanotechnology-based immunoassay that can be used to detect Ebola virus, but unfortunately they are only for lab, such as SPR and QCM immunosensor. And here's a table for a comparison of different Ebola virus diagnosis methods. We can also tell if they are quantitative or not based on this table. Now let's look at the proposed disinfection approaches in domestic wastewater treatment. Sodium hypochlorite and pH adjustment have been found effective in inactivating Ebola virus. Traditional disinfection doesn't work, and no viral inactivation was observed in the first 1-hour test. Following the addition of 5 and 10 mg per liter sodium hypochlorite, no virus was recovered after 20 seconds. This gave us 4.2 log removal of the virus and also a free chloride residual of 0.52 and 1.11 milligram per liter respectively. In addition, elevating the pH to 11.2 was found to significantly increase viral decay over ambient condition. So the first graph here compares the inactivation of Ebola virus using 0 and 1 milligram per liter sodium hypochlorite. The addition of 1 milligram per liter sodium hypochlorite resulted in an immediate free chlorine residual of 0.16 milligram per liter, which inactivated 3.5 log of Ebola virus in 20 seconds. The second graph here shows the persistence of Ebola virus in wastewater at neutral, acidic, and basic pH. No viral inactivation was observed in the wastewater pH of 6.2, but at pH 11.2, the 95% confidence intervals for one log inactivation was found to be 52 to 193 minutes. And here's the 5 things to remember. Thanks for watching.